Good morning and welcome to the Amolyt Pharma KOL webinar on AZP 3601, a potential treatment for hypoparathyroidism. At this time, all attendees are in a listen-only mode. A question and answer session will follow the formal presentations. If you'd like to submit a question, you may do so by using the Q&A text box at the bottom of the webcast player, or by emailing your questions to questions@lifesciadvisors.com. As a reminder, this call is being recorded and a replay will be made available on the Amolyt website following the conclusion of the event. I'd now like to turn the call over to Dr. Thierry Abribat, President and Chief Executive Officer of Amolyt Pharma. Please go ahead, Doctor.
Thank you, Pauline. Good morning, everyone. Welcome, and thank you for joining us today. My name is Thierry Abribat, Chief Scientific Officer at Amolyt Pharma. Next slide, please. Before we get started, next slide, please. Before we get started, we would like to mention that today's presentation and Q&A session may contain some forward-looking statements. Although we are a private company, we believe it is important to comply with all forward-looking statement guidelines as outlined on this slide. Next slide, please. Today, we are pleased to announce the top-line data from the phase 1 trial in healthy volunteers for our lead program, AZP 3601, our PTH analog in development as a potential treatment of hypoparathyroidism. And we're excited to share this positive data with you. Together with me, I have the great pleasure to welcome Dr. John Bilezikian from Columbia University, who I will introduce in a few minutes. Dr. Bilezikian will provide us with an overview on hypoparathyroidism and its clinical needs. Then Dr. Soraya Alas, our Vice President, Clinical Development and Regulatory Affairs, will present our phase 1 data for AZP 3601. Also attending this call are Mike Keller, our Chief Scientific Officer, Patrick Lousteau, our Chief Business Officer, and Lou Arcudi, our Chief Financial Officer, who may intervene during the Q&A session. Next slide, please. For those of you who are not familiar with Amolyt Pharma, we are a clinical stage biotechnology company based in Lyon, France, and in Boston, Massachusetts. Our goal, next slide, please, is to build and develop a portfolio of therapeutic peptides in order to bring new life-changing medicine to patients with rare endocrine-related diseases. With that mission statement in mind, in the last 2 years since the closing of our Series A financing, we have assembled an executive and operational team with established experience and prior achievements in therapeutic peptide and endocrinology, and moved our first program, AZP 3601, into the clinic. Next slide. The last quarter has been particularly productive at Amolyt Pharma with major advances in our product portfolio. Our first program, AZP 3601, is a PTH analog with a higher selectivity and affinity for a specific conformation of the PTH receptor, which has translated in animal models, including animal models of hypoparathyroidism, in a special pharmacological profile with a long-acting effect on serum calcium in the kidney and a neutral impact on bone metabolism. Today, and at the upcoming ASBMR meeting, we are pleased to announce the top-line data from the phase 1 trial in healthy volunteers. We are delighted with these first clinical results for AZP 3601, as they are consistent with the preclinical data, informative, and supportive of a potential therapeutic profile that we believe can address the multiple clinical needs of patients with hypoparathyroidism, including the significant number who suffer or are at risk of kidney disease, osteopenia, or osteoporosis. Based on this positive and promising data, we have initiated the next trial in patients, a 3-month multiple ascending dose study with the objective to report top-line data by mid-2022. Earlier this month, we had also announced our second program, a slide before, please. Earlier this month, we had also announced our second program moving forward into development. Following a successful research collaboration with PeptiDream, our Japanese partner, we have selected AZP 3813, a potent growth hormone receptor antagonist peptide that we intend to develop as a potential treatment of acromegaly as an add-on therapy to somatostatin analog. It is estimated that over 60% of patients with acromegaly still have uncontrolled insulin-like growth factor 1 levels, despite the use of current treatment, including somatostatin analogs. We anticipate developing AZP 3813 as a potential next-generation growth hormone receptor antagonist to provide greater efficacy to a broader range of patients with acromegaly. We are already working to advance these candidates through IND-enabling studies with the goal of initiating the first clinical trial by the end of 2022. Next slide, please. Also, 2 weeks ago, we announced that we have closed the $80 million Series B financing that was led by high-quality investors from the US and from Europe, with also a very strong support from our existing Series A investors. That was great news, just 2 years after our Series A financing. The additional funding will allow to significantly extend our runway to move our 2 existing programs to their next stage of development, including significant clinical milestones, and also explore additional pipeline extension opportunities. So these are exciting times and exciting months of September for us, with the recent advances in our portfolio and the closing of our Series B financing. Now moving to the key topic for today. Next slide, please. It is my pleasure to introduce Dr. John Bilezikian. He's professor of medicine and professor of pharmacology at the College of Physicians and Surgeons, Columbia University. He's vice chair of the Department of Medicine for International Education and Research and chief emeritus of the Division of Endocrinology. He's also director emeritus of the Metabolic Bone Disease Program at Columbia University Medical Center. His major research interests include the clinical investigation of metabolic bone disease, particularly hyperparathyroidism, hypoparathyroidism, and osteoporosis, and has authored over 850 publications in this field. The studies of parathyroid hormone in these disorders, including as a therapy, are known throughout the world as landmark contributions to our knowledge of these disorders. John, we are honored to have you with us today. Please go ahead with your presentation.
So thank you, Thierry, very much for that overly generous introduction. I appreciate being with you all today. I'd like to, in the next 15 minutes, present the landscape as I see it with regard to this very important disease, hypoparathyroidism. Next slide, please. Let us start with normal calcium homeostasis. As shown in this slide, we're dealing with 3 major target organs. You have the skeleton, the kidney, and the gastrointestinal tract. The hormone that we are focusing on today, parathyroid hormone, has a very important effect to regulate these 3 organ systems so that our serum calcium is normal under usual conditions with very little variability. And the way parathyroid hormone does this is to allow calcium to be resorbed from bone and also to conserve calcium as it is filtered at the renal glomerulus. These 2 effects are direct. An indirect effect, shown at the dotted arrow, is through vitamin D. PTH is an activator of vitamin D, and vitamin D directly facilitates calcium absorption from the gastrointestinal tract. So these 3 organ systems under the regulation of parathyroid hormone will allow the serum calcium to return to normal if there is any reason for the calcium to have become low. Next slide, please. Now, in the disease where we want one, next slide, please. There we go. When parathyroid hormone is missing, these 3 organ systems are altered as shown in this schemata. The X over on the upper left-hand corner is the kidney that loses the effect of parathyroid hormone to conserve calcium. In the upper right, the skeleton is no longer a reservoir for calcium as needed because parathyroid hormone is missing. And the bottom left, because of the lack of parathyroid hormone to activate the kidney 1-alpha hydroxylase, there is a deficiency in active vitamin D. And in the lower right, you can see that the absorption of calcium is impaired. So what happens when parathyroid hormone is missing is that the serum calcium falls. Next slide. The disease hypoparathyroidism is thought to be a rare disease as defined by the FDA, with fewer than 200,000 individuals suffering in this country. The demographics are shown here with regard to etiology, gender distribution, and age. Next slide. We recognize this is a rare disease, but we also recognize that it's a very important endocrine disease. Next slide. It is important because parathyroid hormone has pervasive effects on many systems. And when parathyroid hormone is deficient, many of these systems are not working properly. The neuromuscular system is a very important clinical feature of hypoparathyroidism. As you see on this slide, we will deal in a moment with issues of quality of life, renal issues, extraskeletal, and skeletal alterations. Next slide. This comes from our work in which we looked at the SF-36 scale, a very widely used quality of life metric. And the dotted lines at the top of each of these boxes is the normalized score. You can see for each of the 4 physical component summary, as well as for each of the individual mental component domains, those patients with hypoparathyroidism generally score well below normal. And this is true even in those whose serum calcium is controlled with calcium and vitamin D. Next slide. Also very important are the effects of parathyroid hormone lack on renal function. As shown on the next slide, this work comes from Deborah Mitchell at the Massachusetts General Hospital. I'd like you to focus on the graph. This is a study comparing a cohort of individuals with hypoparathyroidism, shown in orange, against a large cohort of normal matched individuals from the NHANES database. And for each one of these age ranges, you can see that the percentage of subjects whose estimated GFR is less than 60 is much greater in those individuals with hypoparathyroidism in comparison to the normal controls. Next slide. This is also very dramatically illustrated by a very recent study in which subjects with hypoparathyroidism, again, were compared with a large database. On the left, with regard to individuals with developing CKD, and on the right, individuals whose CKD has progressed at least one stage. And in both of these situations, you can see that over this 5-year period, individuals with hypoparathyroidism had a much greater incidence of CKD and also progression of renal insufficiency. Next slide. One of the major complications of hypoparathyroidism is not just renal insufficiency, but calcium phosphate deposition. And this takes the form of kidney stones, and is shown here on the left, individuals who developed kidney stones are also shown much more so than in the control group. And on the right, those who developed kidney stones, again, the cohort of subjects with hypoparathyroidism had a much higher incidence over time than the normal controls. Next slide. I mentioned the skeleton as another target organ, and the next slide illustrates a few of the important features. Next slide, please. And this work comes from my group, in which we looked at bone biopsies in subjects who had hypoparathyroidism versus control data. Tetracycline is used as a fluorescent probe, It is picked up when there is active bone metabolism. And as shown on the right, you can see there are very few labels here indicating that hypoparathyroidism is a disorder of very low bone turnover. A disorder of very low bone turnover is likely to lead to abnormalities in bone remodeling, and bone is likely to accrue over time because not only is there a deficiency in bone formation, but there is a deficiency in bone resorption. And again, from our laboratory, next slide, very dramatically is illustrated by micro CT images on the right is a sample of bone from someone with hypoparathyroidism, and on the left is a normal control. You can see that beautiful honeycombing of normal trabecular bone on the left. Now, this is an abnormal phenotype for sure. We actually don't have good data on whether this phenotype can be translated into clinically significant issues, except for the fact that bone density tends to be above normal in patients with hypoparathyroidism. Next slide. So we've touched on a few of these points illustrating that the clinical features of hypoparathyroidism really demonstrate clearly that this is a very pervasive disease. Next slide, please. The challenge is therapy. And up to now, we have relied on calcium. And in this slide, I have added calcium and I've added calcium because we use a lot of calcium to manage these subjects. We also use active vitamin D analogs. Some of us, depending on the situation, will use parent vitamin D, and also depending on the clinical milieu, some of us will also use thiazide diuretics. How much we use of calcium and D and thiazides and in what manner is really part of the art of managing, and it is a challenge, I have to say, manage these subjects on conventional therapy with these supplements. Next slide. And although not always, but often, if we use enough calcium and vitamin D, the serum calcium can be controlled, but it's not always easy. And there are issues that are illustrated on this slide that are not dealt with when we use calcium and vitamin D. For example, the deficiency in renal calcium handling is not corrected. In fact, using large amounts of calcium, I have one patient who has required 9 grams of calcium. And in that setting, hypercalciuria can worsen. And in the context of worsening hypercalciuria, renal function may deteriorate. Next slide. In addition to what I just said, that is, we may have to use large amounts of calcium and vitamin D, the specter of vitamin D toxicity always looms. And again, the abnormalities in skeletal metabolism, although we don't know exactly what that means clinically, they persist. And the bottom line here is that even though patients can be controlled with calcium and vitamin D, and we have shown this and others as well, their quality of life is not improved at all. Next slide. So I call this a central truth. Without parathyroid hormone, many individuals with hypoparathyroidism either are receiving too much calcium and vitamin D, or they are receiving too little calcium and vitamin D. And in both situations, these subjects with hypoparathyroidism are at risk for serious complications over time. Next slide, please. So I also comment that the hypoparathyroidism was the last classic endocrine deficiency disease for which the missing hormone was not available. And on the next slide is a listing of the classic endocrine deficiency diseases on the left and the hormone replacement therapy that we all are familiar with on the right. And now added to this list as of 2015 is hypoparathyroidism, with the approval by the FDA of recombinant human PTH 1 to 84. Next slide. The pivotal clinical trial that led to the approval of recombinant human PTH 1 to 84 is summarized here. This study was a short 24-week study with triple endpoints, namely a 50% reduction in oral calcium and active vitamin D while maintaining the serum calcium. In addition, there have been observational studies that have been as long as 8 years in which those endpoints have been maintained. With this particular product, there is a gradual reduction in urinary calcium excretion, and parameters of skeletal metabolism are increased. Next slide. So the FDA approved recombinant human PTH 184 for the therapy of hypoparathyroidism, and I quote, who cannot be well controlled on calcium and active forms of vitamin D alone, and for whom the potential benefits are considered to outweigh, and then I put in parentheses, the theoretical risk, and this is a rat toxicity of osteosarcoma. Next slide. Since September of 2 years ago, recombinant human PTH 1 to 84 has not been available in the United States, and we do not know when that product will become available again. This is a very short-acting peptide. It does not provide 24-hour control in many subjects, and in fact, some of you may know that when this product was approved by the FDA, it mandated a twice-daily dosing study. We also have to deal with adverse events such as hypercalcemia, hypocalcemia, and because parathyroid hormone has the potential for being a vasoactive agent, we have issues that need to be attended in that regard as well. Next slide. So the emerging therapeutic landscape of parathyroid hormone therapy and hypoparathyroidism is trying to deal with these issues. And the newer approaches include thoughts about convenient delivery systems, more desirable pharmacodynamics, either with prolonged exposure of the ligand PTH or prolonged effect after an interaction with the R0 conformation of the PTH-PTHRP receptor. And let me illustrate that a little bit further with the next slide, which introduces you to the two different conformations of the PTH-PTHRP receptor. The RG conformation on the left is one in which the PTH ligand interacts very, very evanescently, if you will, generating a very brief cyclic AMP signal. Now, this kind of interaction is beneficial when we are speaking about the use of parathyroid hormone analogs for osteoporosis, but it certainly is not what we want in the therapy of hypoparathyroidism. On the right is the R0 conformation of the PTH-PTHRP receptor, in which the PTH ligand interacts in a longer manner. It doesn't necessarily stay for a long time, but it has a prolonged effect, and you can see that intermediate signal of cyclic AMP is more prolonged, giving this interaction or the effects of the interaction a more prolonged action. And this is, of course, what we are looking for in the ideal therapy for hypoparathyroidism, namely a parathyroid hormone ligand that will interact effectively with the R0 conformation of the PTH-PTHRP receptor so as to give that long-lived effect of PTH. Next slide. So the goals of this emerging therapeutic landscape are summarized here. Obviously, we would like the treatment to promptly correct the serum calcium, to provide smooth long-term control of the serum calcium, obviously to eliminate active vitamin D and to reduce calcium supplements to no more than nutritional amounts, as well as to lower the calcium phosphate product. Next slide. In addition, we focused on the kidney because it's a very important target organ. That therapy should reduce urinary calcium excretion, maintain renal function. A very important element, in my opinion, is to improve quality of life. And also the therapy should not lead to an imbalance of bone resorption over bone formation. With all of these goals, next slide, we are also looking at safety first. Such a product should have a favorable benefit-risk profile. Next slide. So I hope this introduction has been helpful to you, refreshing ourselves as to what this disease is, what the challenges are, and how optimistic I think the therapeutic landscape is. Thank you very much.
Thank you very much, Dr. Bilezikian, for this inspiring overview of hypoparathyroidism and its outstanding clinical needs. It is obvious from your talk that this disease affects multiple organs and that the kidney and the bone are very important to consider. And thank you for staying with us to participate to the Q&A session. Now turning to Dr. Soraya Alas, who is going to present the top-line data from the phase 1 trial in healthy volunteers for AZP 3601, our PTH analog that is selective for the R0 conformation of the PTH receptor. Soraya.
Thank you very much, Thierry, and good morning, everyone. Very pleased to present today our data from our phase 1 clinical trial in healthy volunteers. Next slide. We can move next slide. As a short introduction, as it was mentioned before by Dr. Bilezikian, standard of care treatment for chronic hypoparathyroidism, which includes oral calcium supplement and vitamin D supplement, is insufficient to control serum calcium and to control patients' symptoms as well, and to prevent also kidney disease. So the compound AZP 3601 is a novel 36 amino acid PTH analog that is being developed for the treatment of chronic hypoparathyroidism. It has a short half-life and prolonged action, and this profile was designed to maintain serum calcium levels within the normal range, and this in patients, and to reduce urinary excretion of calcium while preserving bone integrity. So we have designed a phase 1 double-blind placebo-controlled single ascending dose and multiple ascending dose study in order to evaluate the safety, the tolerability, the PK and pharmacodynamics of AZP 3601 in healthy volunteers. And we are reporting top-line data here. Next slide. So in terms of mechanism of action, you have been introduced to these two conformations of the PTH-RP receptor. AZP 3601 has increased affinity and selectivity for R0, for the R0 conformation, and a short exposure to the RG conformation. And the specificity is a key differentiator for the compound versus other PTH analogs. It allowed for a prolonged pharmacodynamic effect, especially with respect to serum calcium, and this while having a short half-life, and this is what it is represented in the two schematic on the right, and it has been demonstrated in several animal models, and we are today providing evidence in humans of this profile. Next slide. In terms of study design, as mentioned, the SAD, single ascending dose, and MAD, multiple ascending dose study, were placebo-controlled. In the SAD part, we did test 7 doses of AZP from 5 microgram to 120. The drug was injected by subcutaneous injection in the morning, and a total of 52 healthy subjects did participate in this study. For the MAD, we had 5 doses of AZP 3601 tested in this part from 10 to 80 microgram per day. So the drug was administered once daily. for a total of 14 days, and 50 subjects did participate in this study. Next slide. So for the single ascending dose part, data were partly presented earlier this year at two meetings, the ENDO meeting first, and then the European Congress of Endocrinology. So we are reporting here a summary of the data. Next slide. In terms of safety and PK, a single administration of AZP was generally well tolerated at the doses that were tested. There were no severe adverse event, no serious adverse event. There were no post-dose changes of clinical relevance with respect to safety parameters. And in terms of PK, as you can see on the graph here, there is a dose-dependent PK profile with a short elimination half-life. Next slide, which is expected based on the animal data. In terms of serum calcium, you have two graphs here. The one on the left represents absolute values, and these are absolute values of serum calcium over the day. So we did we had several sampling time points over the day in order to follow the profile of the serum calcium. This is on the left. And then on the right, you have area under the curve. So basically what's happening during the day. So this is during 24 hours. You can see on both graphs that clearly there is a dose-dependent effect. There is an increase in mean values versus placebo, which is the gray line. And you can see also that the increase in these mean values was sustained for up to 24 hours at the doses of 40 as well as 60 microgram. So basically the orange and red lines, and for up to 48 hours, which is not shown here in the graph, but at the highest dose of 90 and 120 microgram, we had sustained effect over 2 days. And then mean values returned progressively to baseline values. Next slide. Now looking at endogenous PTH, there is a clear, you can see that on the left, a clear dose-dependent decrease in mean endogenous PTH values, and this is expected. As you increase serum calcium, you decrease serum PTH. You can see also that the mean values remain lower after 24 hours at the highest dose. So basically the green and the blue line, 90 microgram and 120 microgram. And what you can see on the right, which is very nice, it's a correlation between the two pharmacodynamic parameters, serum calcium and PTH. And you can see that endogenous serum PTH values were inversely correlated with serum calcium, which is expected. Next slide. So in terms of urine calcium, as it was mentioned, it is a very important parameter to look at, especially when treating patients with chronic hypoparathyroidism. We can see here, and we have two sets of data. First, on the left, the 24-hour urinary calcium. So this is the collected over 24 hours. That is corrected for creatinine. And you have on the right, what we call the fractional expression of calcium, which is basically the urine calcium values that are adjusted with the serum calcium values. And both graphs, both sets of data are telling that there is no increase in urinary calcium expression, despite the fact that we are seeing, and especially at the very high dose, marked elevation of serum calcium. So this is suggestive of reabsorption of calcium, and we're going to see that in more details with the MAD data. Next slide, please. In terms of conclusions, so a single administration of AZP 3601 at doses up to 120 microgram was well tolerated, associated with no safety concerns. PK and PD data are in line with the target product profile of the drug, short elimination half-life, and sustain dose-related calcemic response over at least 24 hours. And this is what we want to achieve in patients in order to be able not only to control serum calcium, but also to control symptoms. So this sustained effect of serum calcium was associated with a dose-dependent decrease in endogenous serum PTH, as expected, and very importantly, was not associated with any increase in urinary calcium excretion. Next slide, please. We can go now over the multiple ascending dose data. Next slide. So in terms of safety, generally, it was well tolerated with no safety concerns. We had no SAEs, no severe adverse events. The most frequent adverse events that were noted were considered to be related to hypercalcemia. We had nausea, headache, somnolence in some patients. And these adverse events were observed with dose-dependent incidence and severity. They were observed at 40 microgram per day and higher, and were more pronounced in 2 subjects at 60 microgram per day. But we didn't have any, at 80 microgram, we didn't have any subject that did not tolerate the dose. No post-dose changes of clinical relevance were observed with respect to safety parameters, consistent with the SAD data. Next slide, please. So in terms of serum calcium now, what you can see on the left is the mean values over the treatment period, plus also we have added here the follow-up period. So basically people were resident in the phase 1 unit during 14 days, and they stayed for a couple of days following that, and then they were called again, they were tuned to the phase 1 unit for some follow-up sampling. What the graphs here is showing is that there is a dose-related increase in serum calcium that is quite rapid. You can see that very quickly after the start of the treatment, that this increase is sustained for the duration of the treatment period, which is 14 days. And when you look at day 15, which is the day following the last dose, you can see very clearly that the increase in serum calcium is maintained after this last dose, confirming the long-acting effect of AZP 3601. Now, when we look at the 2 graphs on the right, this represent what is happening during the day on day 7 and day 14. We had on these 2 days several sampling time points in order to look at the serum calcium profile. And what you can see very clearly on those 2 days is that the increase in serum calcium is maintained throughout the full 24-hour period. And this is also something that we want to achieve in patients, to be able to have serum calcium that is controlled over the day in order to control symptoms as well. Next slide. In terms of serum PTH, as for the SAD part, there is a decrease that is also rapid in mean serum PTH versus placebo, which is dose dependent, very consistent with the rise in serum calcium. And what you can see, especially at the highest dose, is that PTH remains suppressed throughout the treatment period. Next slide, please. Next slide. Thank you. So when we look now at phosphorus and in particular the calcium phosphate product, this is something that is important to look at when treating patients with chronic hypoparathyroidism. As it was mentioned in the previous presentation, this is a therapeutic goal to lower calcium phosphate product in hypoparathyroidism patients. So while there is an increase, this is what you see on the left in serum calcium, you can see a dose-related decrease in serum phosphorus, which is dose-dependent, and this is the graph in the middle. And finally, the graph on the right, you can see very clearly that there is also a decrease in this calcium phosphate product. So this is what it is expected to be observed as well in patients. Next slide. Let's have a look now at the urinary calcium pharmacodynamics. And you have on the left a graph that represents the fractional expression of calcium, urinary calcium. You have also this this purple line that represents the upper limit of normal, which is 2%. So you can see very clearly, and this is over the treatment period of 14 days, plus you have the follow-up also represented here on this graph, that there is no increase, basically no change. All what you can see is non-significant change, despite the increased serum calcium levels that were mentioned previously. At the dose of 80 microgram, you can see a slight increase versus baseline, and this is largely due to one subject who had frank hypercalcemia, so basically values above 12 milligram per deciliter, which is really high as compared to other subjects who were around 10.6 or 11. And these values were observed from day 7 to day 14, and this may explain this increase in urinary calcium that we see specifically in these patients. The graph on your right represents the serum calcium as well as the urinary calcium. So both data here on one graph. Basically, the orange line is increasing, which is the serum calcium. And what you can see is that despite this increase in serum calcium, there is no increase in urinary calcium, which is the purple line. Next slide. Now, with respect to bone biomarkers, we've mentioned that it is very important to look at bone in this patient population. We have measured CTX, which is the catabolic bone biomarker, and PYNP, which is the anabolic bone biomarker. So the two graphs here show very clearly that following 14 days of treatment, there is no relevant changes at any of the doses that were tested, reflecting what we call a neutral effect on bone turnover, which is expected based on the mechanism of action of the drug. Next slide. So in terms of conclusion, repeat administration of AZP 3601 for 14 consecutive days was generally well tolerated. PD data were in line with the long-acting effect of the drug. This is what we want to achieve. Rapid and dose-dependent onset of increase in serum calcium on day 1 that was sustained and stable, maintain also up to 24 hours after the last dose, so after day 14. This effect was associated with a dose-dependent decrease in endogenous PTH, which is consistent with the rise in serum calcium, as expected from the mechanism of action. In addition to that, no increase versus baseline in urinary calcium at doses up to 60 microgram, despite increased serum calcium, and no change in bone biomarker at any of the doses that were tested, reflecting a neutral effect on bone turnover, as expected, again, from the mechanism of action. Next slide, please. So what we would like to highlight here is that the patient population who have chronic hypoparathyroidism is an heterogeneous one. Two organ systems are of importance to preserve from a safety perspective. We mentioned, and Dr. Bilezikian mentioned, the kidney. This is important when treating those patients not to have hypercalcemia. We know that a significant proportion of them have already kidney disease. chronic kidney disease. So this is important when treating these patients to take care of the kidney. The second organ system who is of importance also from a safety perspective is the bone. Why? Because there is also a significant proportion of patients who are already fragile with respect to their bone because they have osteopenia or osteoporosis. And also, very importantly, the great majority of patients are women, and women at their peri or postmenopausal, and those women are at risk of developing conditions such as osteopenia and osteoporosis as well. Next slide. So having that in mind, the data presented here, we believe that they further support the development of AZP 3601 as a potential treatment for chronic hypoparathyroidism. We have seen the increase in serum calcium, and this is expected to translate into elimination of calcium and vitamin D supplementation in the patient population and improvement of symptoms. We have seen also that data indicate reabsorption of urinary calcium. So this is also important to have in patients. This is a marker of the prevention for kidney disease, a very important key and a key therapeutic goal in this patient population. And as well, no effect on bone biomarkers after 2 weeks of treatment, indicative of a neutral effect on bone metabolism. And this also may be a key safety advantage, as we mentioned that this patient's population may be fragile as well with respect to their bone. Of course, all that is based at this point on data from healthy volunteers, and patients' data will tell more. And we are currently conducting a multi-center 3 months multiple ascending dose study in patients with chronic hypoparathyroidism. This is currently ongoing, and we hope to share data with you in the near future. So I would like to thank you very much for your attention.
Thank you, Soraya.
Thank you
for your presentation. We are particularly pleased with this data as they fulfilled our expectation in many ways. In healthy volunteers, we observed a prompt, potent, and sustained effect on serum calcium, lowered calcium phosphate product, a good and effective control of urinary calcium excretion, indicating enhanced calcium reabsorption by the kidney, and importantly, a neutral effect on bone metabolism, which means no imbalance between bone resorption and synthesis. As you heard from Dr. Bilezikian in his concluding remarks, these are important features of what is expected from a PTH treatment for hypoparathyroidism. In addition, the data suggests that the overall impact of AZP 3601 on calcium metabolism is primarily driven by its effect on kidney, because it has very little effect on the bone, which we believe could ensure a long-term sustainable effect. As mentioned, we have initiated the next trial in patients and look forward to reporting mid-next year how this profile translates in terms of safety and efficacy in patients. So this is the end of the presentation. The presentations. Back to you, Pauline, for the Q&A session.
Thank you, Thierry. At this time, we'll be conducting a question and answer session. If you would like to submit a question, you may do so using the Q&A text box at the bottom of the webcast player, or by emailing your questions to questions@lifesciadvisors.com. Please hold while we pull for questions. I'll now turn the call over to Glenn Garment of LifeSci Advisors to moderate the Q&A session.
Thank you, Pauline. Our first question this morning is for Dr. Bilezikian. Based on your broad clinical experience in the field, what are the key data points from this phase 1 trial in healthy volunteers that you found particularly relevant for AZP 3601 as a potential treatment for hypoparathyroidism?
Well, thank you for that question. There are a lot of interesting points to highlight, but I would say the one that I'm focusing on more than the others, perhaps, is the apparent effect on renal function in terms of renal calcium handling. This is really one of the major problems we see in hypoparathyroidism. Diminishing renal function, kidney stones, nephrocalcinosis, and this product, and Soraya nicely highlighted it, as the serum calcium is rising, the fractional excretion of calcium is not going up. In fact, it's staying a level, which is quite amazing, I think, from a physiological point of view. So if you wanted me to point out one of the most important, there are other interesting facets, but I would focus on the way this drug seems to be affecting beneficially renal calcium handling.
Okay, thanks very much, Doctor. Our second question, Theory, I'll direct this to you to decide who should answer. This looks like clear and promising data for AZP 3601 and an interesting product profile, but could you let us know why you believe that a neutral effect on bone metabolism would provide a safety advantage to these patients, whereas it has been reported that they have more bone than the normal population?
Yeah, I'm going to take this one. When you develop a new potential drug, you have to know the mechanism of your drug and how it can affect the underlying biology of the disease. And importantly, you have to know the patient population that you want to treat as well. And with regards to bone, it is true that patients with hypoparathyroidism have a little bit more bone than the normal population, how Dr. Bilezikian mentioned in his presentation, but this is in average. And this is associated with an impaired quality of the bone. There is no evidence that this added quantity of bone is protective against fracture. Actually, the available data suggests that hypoparathyroidism patients have approximately the same fracture rates with aging as a normal population, no more, no less. In addition, also the mean bone mineral density seems to be higher. There is a distribution, right? And a significant percentage of patients have low bone mass when compared to the other patients, the normal population of the same age. So they have negative Z-score as well. So in those patients in particular, we believe you want to be cautious and make sure, as stated by Dr. Bilezikian, not to induce excess bone resorption over bone formation. So that's why we believe that the PTH product with a neutral impact on bone metabolism, which is what, at least so far, AZP 3601 seems to be doing, could be well suited for all patients, including those with fragile bone, osteopenia, and osteoporosis.
Thank you, Theory. Our next question comes from John Woolabin from JMP Securities. And again, this is for you, Dr. Bilezikian. When recombinant PTH was available, how were you utilizing it in your patients versus supplementation alone?
When PTH 184 was available, and if patients met the strict criteria that the FDA had pointed out, namely those who could not be well controlled on, and I'll put reasonable amounts of calcium and vitamin D, that is the population that I would used a recombinant human PTH 184 for. Now, the question also needs to be qualified because I have a very highly referred practice, which is to say patients with hypoparathyroidism who come to me and my group tend to have hypoparathyroidism that is very difficult to control. So while the local endocrinologist may say a minority of their patients with hypopara met those criteria, in my experience, the vast majority did. So I would use a recombinant human PTH quite commonly, and I won't go into the algorithm that we used by which we would reduce the D and the calcium, but of course we're very familiar with how to do that. So I became very familiar with it, and I have to say that the landscape of this disease was changed by the advent of this first PTH product. And obviously we look forward to more progress because of the need to improve on that first step.
Thank you, Doctor. Thierry, our next question comes from Leland Gershell from Oppenheimer. When will we see initial data in hypoparathyroid patients? Would we expect a different effect on bone in patients versus healthies?
Yeah, this is a forward-looking question. So as I said in the introduction, our objective is to have the data in patients by mid-2022, in a few months from now. So we believe we'll have a similar effect on the bone in patients versus healthy, but we have to see the data first. It's going to be a longer-term treatment, up to 3 months of treatment. What we have seen in animal models is that in animal models of hypoparathyroidism, when you remove the parathyroid gland and create a state of PTH deficiency in animals, we also saw a natural impact on the bone metabolism. But of course, we have to generate the data in patients first.
Thanks, Thierry. Next question. Can you explain what conclusions we can draw from your results on urinary calcium excretion, if any?
I think that Dr. Bilezikian just made a very nice statement on the significance of the data from this phase 1 trial in healthy volunteers. The control of urinary calcium excretion is a very important therapeutic goal, as these patients are particularly prone to kidney disease. I will not come back to that. The difference between patients and healthy volunteers is that in patients, some have elevated urinary calcium, some have normal urinary calcium levels. The goal of the treatment would be to decrease, especially those who have elevated urinary calcium, and keep those who have normal range, within the normal range. What we hear from the endocrinologists also is that try to lower urinary calcium, even in those who are at the threshold. We hear the lower the better, and this is hopefully could be associated with an improvement in kidney function as well, or no degradation of kidney function that is observed in those patients. So let's see what the data in patients, but we are very pleased with the data we have in healthy volunteers. I don't know, John, if you want to add anything to that.
Well, Thierry, I agree. The other point I would make is that we have focused in this disease a lot on the stones and the calcium deposition. But you made the point, and I did as well, renal function is really important. And the data, the recent data published looking at cross-sectional 5-year data, that renal function itself, and I believe this is the calcium burden that is responsible for the reduction in renal function. So we really need to emphasize the point, not only the risk of calcium deposition, but also the functioning of the renal system. And these two elements would theoretically be advantaged by a drug that had a major effect on renal calcium excretion.
Thank you.
Okay, thank you. We have a follow-up now from Mr. Gershell at Oppenheimer. Again, Dr. Bilezikian, this is for you. Concerns have been expressed by some that long-term PTH replacement could overcorrect bone mineralization and put this below the lower limit of normal. how long would we need to see clinical data run out to be comfortable that this is not a significant risk?
Yes, so a good question. There are early parameters and there are later parameters. The early parameters, of course, the bone turnover markers, and Soraya has nicely introduced the markers that we use. The CTX reflects bone resorption, and the P1NP or the osteocalcin or the bone-specific alkaline phosphatase would reflect bone formation. Now, those are early parameters, and I would expect to see signals within a very short period of time. Tracking those over time will give a better sense of whether the change or no change is maintained over time. The longer index that would give us more specific information would be to track bone density. And the earliest time point to see a change in bone density would be, I would say, 6 months if the effects on bone metabolism are, I would say, strong, if I could put that word in quotes. And certainly by 1 year, one would see effects on bone density. So this is an area that needs attention and should be tracked for whatever drug is being developed.
Thank you, Doctor. We've now arrived at the last question, and Thierry, I believe this would be for you and the team. Thanks for an interesting presentation and congratulations on promising data. How do you view the commercial potential of 3601, and what do you anticipate your commercial approach will be?
We are not exactly there yet in terms of a commercial launch. We have just produced the data in patients, and we look forward to accelerating the development as much as we can when we have the initial data in patients, because we believe we make our best to bring that product to the patient as fast as we can. But I will let Patrick Lousteau, our CBO, answer the commercial part.
Sure, Thierry, thank you. Yeah, I'd be happy to answer the question. We have actually conducted thorough market research, both in the US and the EU. And maybe from that research, I can use and talk about two main conclusions that would probably answer your question. The first one would be that based on this work, the hypoparathyroidism market is actually very large, owing both to the high prevalence for a rare disease, but also to the high level of unsatisfaction with a standard of care treatment. And based on our work, that leaves room for several products to coexist and successfully fulfill all the different unmet needs. The second point is that we have actually realized that the hypoparathyroidism population is very heterogeneous. You heard that from Soraya. There are patients with very distinct clinical needs that go beyond the immediate need for maintenance of serum calcium in the normal range, elimination of vitamin D and calcium supplementation, and control of symptoms over 24 hours. And these distinct segments may be better managed actually by different product profiles, and one size may not fit all for this condition. As a matter of fact, our market research data is giving us a solid indication that for several of these patient segments, AZP3601 actually was seen as a very strong contender and even a preferred product based on the profile that you have seen today in the phase 1 results. And in particular, and as you heard from both Dr. Bilezikian and Dr. Alas, I can highlight the sizable groups of patients who have developed kidney disease, which is about 26%, the ones who have developed osteopenia or osteoporosis, which is close to 20%, and all those who are at risk of developing these comorbidities. And for all these patients, our physicians in the market research actually favored AZP3601. So this doesn't mean that we are going to focus only on these patient groups, as our product has been designed to, and it is actually also being developed for the potential treatment of hypoparathyroidism for all the patients. But we think this is a good starting point. And all in all, these market research data are quite encouraging for the commercial potential of AZP3601. of course, if the product profile is confirmed in the next trial in patients.
Thank you, Patrick. This marks the end of the Q&A session and therefore concludes this morning's webinar. We hope you found it to be informative. Thank you, Dr. Bilezikian and Thierry and the team, and everybody have a good rest of the day.
Thank you very much.